The next talk is computational approach to detect chimeric transcripts with paired-in RNA-seq experiments, FusionSeq, and this is with Andrea Sobner, and he's also from Yale University.
Good afternoon, everyone, and thank you very much, the organizer, for inviting me to present my work here today. This work has been done in close collaboration with Yale University and Weill Cornell Medical College. And it regards a particular application of RNA sequencing, a paired-end RNA sequencing, to detect chimeric transcripts. So, as you probably know, there are a lot of advantages in using next-generation sequencing. And in this particular application, one of the advantages that is more important is the fact that you can do unbiased genome-wide type of analysis. But of course, along all the advantages, there are also some challenges. Those are equipment that generates a huge amount of data that requires computational tools to handle, which is good for the bioinformaticians. And even if the cost of the sequencing itself is going down, and probably will go down in the future, there are still additional costs that probably will be increasing in the future, like the cost of the downstream analysis to figure it out, what are the most important elements that we discover using these machines. And here in this cartoon, we actually represent this as the orange bar. So in the future, 10 years from now, probably the highest fraction of sequencing experiments might be the downstream analysis, as well as also the experimental design, because you might use these machines to address more complex type of experiments and biological questions. RNA, in particular, in this talk, I'm going to talk about RNA sequencing, which is the application of next-generation sequencing to transcriptome. And what you could typically do with RNA-seq is standard analysis like expression measurement at different levels, at the gene level, at the exon level, at the transcript level. At the same time, you can also look for investigate alternative splicing. You can do transcript reconstruction de novo, for example, discovery of novel expressed regions, all in one experiment, and many more other types of analysis. In RNA-seq, there are some challenges that are, as I said, common also in every next-generation sequencing experiments, like storing data, processing, and sharing. But in particular, if you also deal with human subjects, there are also some privacy issues, because using sequences might be sufficient to identify or even characterize a person. So in this context, we developed a set of tools, including data format called map-free format, and the tools are called RC tools, to deal with all these issues. So the data format is able to capture the alignment of short, long, single-end, and paired-end reads. It can generate summary data, which are very useful, especially now that sequences are getting longer and longer, and the throughput, the output of the machines is also increasing. But more importantly, it enables the separation of the public information, the alignment, the things that can be shared, from the private information, the sequencing of the individual.